Alright, so we are going to start looking at our 1B slides. Remember that these slides can be found inside your 1B slides, that orange folder in our Schoology. We're going to always be going through those, using those, so these looms come directly from there, so you can pull those into OneNote, into a Google Doc, whatever you want. You want to take these, your notes completely separately, totally up to you. But we are going to be working through those 1B slides. So, without further ado, the start of 1B, Introduction to Biotechnology. So, let me make myself small. We'll put myself over there, and let's get started. So, short little one for our first one, kind of just like a real introduction to an introduction kind of thing. So, come on. All right, so biological hierarchy. Remember this? Do you remember this stuff? So here we have an Amoeba Sisters video. I'm not going to play it in this loom. You can definitely go back and look at this. So it's going to talk to you about different cellular organization, organization within an ecosystem, so kind of like all the different biological levels. So make sure that you watch this, make sure that you're okay with this vocabulary. If anything of this is like, where did this come from? You need to come in to office hours. You need to ask questions. It's okay that you have questions. Be honest with yourself. Realize that you have the questions. Don't be embarrassed to ask. I don't care. I only care that you don't ask questions or that you are confused and you're not asking them. So that's not good. So watch this video. It's Amoeba Sisters. I usually use Amoeba Sisters completely as an introduction because we go so much higher than what they go into. So Amoeba Sisters. So organisms and their components. Remember, we had this definition of biotechnology way back when we first started. It was one of the first things that you did within a digital interactive notebook on slide 4, where you had to sort out the vocab terms. So what we said, it was a way to the study and manipulation of organisms, their cells, their tissues, their components for human needs. So remember that when we work with these organisms, we have to work either with the organisms themselves or with components of those organisms, cells, tissues, cultures, things like that. So all of those components become what is known as raw materials. So that is all of the products, all of the components of the product of what you're making. So take the SARS-CoV-2 vaccines, like part of those raw components is the actual virus, and the virus was sent out to all of these companies. Even when you're talking about the animal trials where they gave them the vaccine, they need to challenge it with introducing them to that to the SARS-CoV-2 virus. Some of the particles, the raw materials, are also the spike proteins. It's the adenovirus vector. So it's any of those raw materials that make up the product that you are looking to produce. Another example are antibodies, like the Regeneron, the Eli Lilly products. So those are the antibodies that are getting produced. Cells, your best friend. Oh, you have not met your best friend yet. Your best friend's E. coli. And when we get into our lab, we make E. coli all the time. So when we're learning how to gram stain, you learn how to do it on E. coli. When we're learning how to streak a plate, you learn it with E. coli. When we learn how to do simple stains, even more simple than the gram stain, it is with E. coli. So E. coli is our best friend. But like everything else within this year, yes, E. coli can make you sick. So we want to make sure we have always, I had kids when everyone was like, oh my God, we need to disinfect. I had old students messaging me about what products we use to clean in here. So cleaning and disinfecting and making sure that we are in a sterile environment, as sterile as can be, is something that we do all the time in this classroom. So part of that is because you get used to working with your best friend, E. coli. Other pieces that we are considered raw materials are the multicellular organisms. So it is the corn, the strawberries, things like that. So it is the entire organism, not just a component of it. Sorry, Mr. Ray's is like making noises. I'm like, what is he doing? It's Ray's. So what are your questions? You need to ask questions. What we were talking about before with the Amoeba Sisters, but now this is even bigger. Bigger than just like questions on content. You need to think of potential problems, potential issues, potential complications in using these materials and using and conducting these experiments. So if your goal is to use one of those raw materials, I want you to think right now about what you want to understand to be able to accomplish, quote unquote, accomplish biotechnology. So let's go back 1 second and look at these raw materials. So we are talking antibodies, we are talking cells, whether it be E. coli, yeast, liver cells within our own cells, stem cells, up to full multicellular organisms, corn, strawberries, animals, plants, us, full multicellular organisms. My question to you first is, if you want to use those raw materials, what do you want to know about those raw materials before you do anything, before you experiment on anything, before you design up any protocol at all? I want you to think right now. Pause this, literally pause this, and I want you to spend some time and think about, what do you want to know? What questions do you want to ask before doing any of those experiments? Think about it. Pause me. No, seriously, pause this. Answer those. Okay? Think about that activity. I'll wait. All right, so coming back, some of the things that you might want to think about are the different characteristics of life. So think, what is their genome like? Has the genome been sequenced? If not, can we sequence it? How expensive is it to sequence it? How expensive is it to extract it, to preserve it, to utilize it, to perfect it, to get it so that you really can understand the genomic level of that organism? What is unique to that organism? Where does it live? What interactions does it have with its ecosystem? Is it predator, prey, friend, foe? How does it fit into its niche? Okay, all of those different things. You also want to understand any of the structures that compose that organism. So especially if it is multicellular, you need to know its genomic components about every single type of cell or every single interaction that it might have. If you're making a pharmaceutical product, you want to know how that is going to happen or how it's going to work on the entire organism, not just the organ of interest that you are looking to make a drug product for. So you need to know how it's going to work within everything. And to understand that, you need to understand how it is the entire system is made up. So, ready? Here is a activity for us. So your job is to develop a pesticide-resistant soybean plant. Not plan, plant. What do you want to know about that soybean plant? Okay, what do you want to know about that plant? Again, what do you want to know about that plant before you start designing anything, before you start experiments, before you start tinkering, before you start playing, before you start making any sort of drug target or treatment protocol for that soybean plant? What do you want to know about it first? Think about it. Answer those questions. Pause this. Pause this. Stop. Pause. What do you want to know before you move forward? So, hopefully you paused that, you thought about that, you maybe went back to that slide right beforehand to look at different things that we want to learn from before about structures and genomic structures and protein structures and how it interacts with the environment. All of those components would have been things that you wanted to learn about that soybean plant before you designed anything. So, next piece, this video. This is another video that I would like you to watch. This video should again be a major review. Should, operative term. If you have any questions with this video, any problems, any like, I don't understand this component of this video, you need to see me so that we can go over this together, okay? again, we're talking basics, but before we can actually get into things that we're going to be talking about, you need to understand those basics. So if you are with me in class, we might do this over our, over the, I guess, Zooms. We're going to see how this works. Maybe we'll figure something out, so maybe we can play Kahoot all together. But in terms of what's going to be due within the DINB for this little mini unit, if we go over to our 1B DINB, we are going to be working on slide 5. So I need to scroll back up here. Okay, so hopefully on slide 3 there's our little introduction to our unit. Please watch this. Okay, please watch this as a little introduction of what we're going to be learning about this unit. In slide 4, okay, on slide 4 we are looking at biological hierarchy. I might rearrange these. We'll see. So they might be in a little different order when you see this. I think I might move the table of contents to be before biological hierarchy. That does make a lot more sense. I don't know why it's there. Silly me. But so with this, you are looking at what is biological hierarchy. So you're answering this question that's right up here. And you are going to think about now plans for your best self. What do you want to help you to focus on this class? So you guys have a lot on your plate with, applications and SATs and everything else that goes along, plus all your other classes. Kind of think about this as a time to make a little bit of a promise for yourself. How are you going to focus on this class specifically? And then you're also going to answer this. How are you going to set yourself up for success within the school year as a whole? So what are you going to do to help yourself there? And last but not least, what I would like you to do here, this was supposed to be slide 4. Yeah, I don't know why these moved. So we're going to move these back up before this even gets shared with you. Hopefully that moves. Yes, perfect. So on this slide, which is your table of contents, which is now on slide 4, what you're going to do here is you're going to be listening to these videos. And these videos talk about different ways to take notes, different ways to approach study habits, all those different things. And I want you to actually watch them, not just for this class, but for all of your classes. And I want you to think about what of these would work for you. Also, what of these would not work for you? So really starting to get a little bit of introspective on your personal study habits so that, these questions now on these notepads on slide 5 make a little bit more sense in this order. I don't know why that got moved. Oh well. So you're going to put a little screencast right here showing you a little bit of your introspection. You can see within our table of contents all the different things that we're going to be doing this unit. So kind of a busy unit, kind of not that bad. So first piece is done. So a little bit of an introduction. We saw the importance of different types of raw materials. We talked about what raw materials were. We talked about different questions that we need to ask with those raw materials, different components that make them become very important, especially when you're thinking, planning, doing experimentations, back to that legal and safe lab slide deck, so that when you want to actually make up your idea, make up your experiment, that you have all of those questions answered as part of your experiment. So that is the end. As I sneezed, sorry, bless me. So as you watch the 2 videos on 2 and 6, make sure that if you have any questions that you are asking me, and I will see you next time. Take care.